I want to confess something. I'm more microbial than I'm human. I have more microbial cells in my body than I have human cells. I have microbiota. We have microbiota. We have communities of microbes that assemble beginning at birth on our skin, in our mouth, in our gut. The largest community in our gut. Tens of trillions of organisms representing all three domains of life on the planet. We never dine alone. We have trillions of friends. In addition to these communities of microbes, microbiota, we also have microbiomes. Vast collections of microbial genes in the genomes of these organisms that together form a microbiota. We live in peace and harmony in health with these organisms. Our encounters with microbes are not framed typically in warlike terms, but rather in a co-evolved, mutually beneficial living together or symbiosis. This recognition of ourselves has been spawned in part from the same revolution in genome sequencing and computational biology that brought us our Homo sapiens genomes. This perception of the microbial side of ourselves is giving us a new view of our individuality, a new sense of our connection to the microbial world, a sense of the legacy of our personal interactions with family and environment early in life. It's causing us to pause and consider that there might be another dimension to our human evolution, one that's occurring much more rapidly than that portrayed in our Homo sapiens genome, one that's occurring at the level of our microbiomes, reflecting our different lifestyles, our changing way of eating, our travel, all those sorts of things that are lumped under the term Westernization. This perception of ourselves as a sublime combination of microbial and human cellular and genetic parts is also giving us an opportunity to look at some of the most vexing and some of the most challenging global health problems. And that's the subject of my conversation with you today. Malnutrition, or undernutrition specifically for today's conversation, is a devastating problem affecting children and women throughout the world. A problem that's affecting not only the capacity of people to achieve their full potential, but one that is manifest by suboptimal growth, by abnormalities in neural development, by immune dysfunction. We measure undernutrition in children currently by taking a view of how tall they are as a function of their age, how much they weigh as a function of their age, and comparing these parameters, these anthropometric parameters, to a reference control population of children designated by the World Health Organization as healthy growers. Now, the magnitude of the problem of childhood undernutrition is overwhelming. In 2011, over 150 million children under the age of 5 were stunted. Over 100 million were underweight. 50 million were moderately or severely wasted. Now, undernutrition isn't simply due to food insecurity, a lack of food. It's thought to reflect a very complex intra-generational dynamic as well as intergenerational dynamic. And people have been spending an awful lot of time trying to understand how certain aspects of how we live, how certain aspects of the food that we consume might increase the risk for undernutrition. But we haven't found a single cause yet. It's this complex dynamic. And I'm going to add one more factor to our thinking about undernutrition today. and that's to turn to this terra incognita that's becoming more cognita, our microbiota, and ask whether the microbiota is causally related to risk for and the manifestations of childhood undernutrition. So this is Tanya Yatsenenko and Satish Subramanian, very talented graduate students at Washington University, and together with their colleagues, they've been sampling the gut microbiota of people living on 4 different continents, 4 countries, embracing 4 very different cultural traditions and lifestyles. And what they've found is that it takes about 2 to 3 years for our gut microbiota to assemble into an adult-like configuration following birth. During this period of time, they can see through DNA sequencing of members of the gut community a change in the representation of microbial genes in the microbiome. There is an identifiable program of maturation of the microbiome, and it involves genes that are critical for healthy growth, such as genes that are involved in the synthesis by microbes of vitamins that we need, for processing certain components of our foods that we're not able to do just based on the dining utensils encoded on our human genome. They also see, and we're also seeing, that a mother's microbiota changes dramatically around the time of birth and following birth. Father's microbiota don't change during this period of time. I won't comment further about that. But this change in microbial community structure, guts of mothers, we think is very important for ensuring transmission of microbes from mother to child. And we think about the infant maternal dyad as key to this transfer across generations of traits encoded in the microbiome that are critical for health. Family members share microbes. Their gut microbial communities are more similar to one another than unrelated individuals living in separate households. But a curious thing is that the gut communities of identical twins are not more similar to one another than the gut communities of fraternal twins, implying that early environmental exposures are key determinants of the types of microbial communities that we assemble later in life. And by the way, once we acquire these microbes, they appear to persist for very long periods of time, decades, maybe throughout the course of our lives. And if that's true, that means that our early exposures to microbes have long-lived effects on the features of our biology, our physiology, our metabolism, our immunity, that determine our health status and our interpersonal variations. So you can ask, if there is a program of assembly of microbial genes in our microbiomes following birth, and if this program involves genes that are critical for generating nutrients like vitamins that are critical for growth, processing components of our diets that otherwise couldn't be accessed with the utensils encoded in our human genes, what happens if this program is perturbed? So I'm going to ask a few questions about the microbiome in the context of undernutrition that actually could be framed in the context of other diseases. Are there identifiable associations between the shape, the structure of a microbiome and a disease, in this case, moderate or severe undernutrition in children? Severe acute malnutrition, SAM. If there are correlations, they may give us new ways of diagnosing or classifying the disease. They may give us new ways of understanding the origin of disease. What happens with therapy? Now, there's been a remarkable innovation in the treatment of children with severe acute malnutrition, and that's to give them ready-to-use therapeutic foods, one of which is shown here. It's a peanut-based therapeutic food, or RUTF. It consists of peanuts, of powdered milk, micronutrients, and it really has dramatically reduced mortality due to severe acute malnutrition. It promotes weight gain, but it doesn't cure some of the long-lived effects of the experience of undernutrition. Neurodevelopmental problems, reductions in IQ, defects in immunity, poor responses to vaccines. So is the microbiota reconfigured with these therapeutic foods? And is the reconfiguration durable once these foods, which are typically given for a few weeks or a month until the child begins to gain weight again, is the reconfiguration durable? And is the reconfiguration similar across many different geographical locations, or are these effects quite local? So in order to understand some of the possible answers to the questions I've just proposed, the Bill and Melinda Gates Foundation has funded a project. We and our collaborators, which represent many different countries and many different academic institutions, are looking at the microbiomes of children with undernutrition. I'm going to tell you about one such study. It involves Tanya Yatsenenko, who I just introduced you to, as well as an extraordinary emergency room physician here at Washington University who spent at least a half a year each year for the last 2 decades establishing an infrastructure for the identification and treatment of children with undernutrition. And that person is Mark Maneri. And what Mark and his colleagues, together with Tanya, have done is to recruit 317 twin pairs living in 5 rural villages in Malawi, follow them through the first 3 years of life, and look at their health status and whether they developed severe acute undernutrition. And there are 2 forms of severe acute undernutrition. One, kwashiorkor, which has a much more abrupt onset. Another one, marasmus, which is much more progressive, takes a longer time to evolve. Among this group of 317 twin pairs, over the course of the first 3 years of their lives, 50% of the twin pairs remained healthy, but an amazing 43% became discordant for moderate or severe undernutrition. One co-twin in the twin pair remained healthy, the other developed undernutrition. And there was no significant difference in the incidence of discordance between monozygotic or identical twins and fraternal or dizygotic twin pairs, which points to the important role of some environmental exposure. Now, the health care standard in Malawi is that if a twin pair is discordant for undernutrition, both co-twins are treated with RUTF. There's concerns about maternal food sharing, that the mother would devote all the food resources to the unhealthy twin and put the healthy twin at risk for disease. So we have a perfectly controlled experiment within the context of a discordant twin pair. Study their microbiomes before, during, and after treatment, and compare the 2 co-twins, and then do the same thing across multiple twin pairs. And the results showed that the co-twin in these discordant twin pairs who have severe acute malnutrition have an immature-looking microbiome, and that the ready-to-use therapeutic food, which is applied for just a few weeks or month, is not sufficient to repair that immaturity. It does so just transiently. But these are just associations. They don't necessarily tell us that there's a causal relationship between the microbiome and severe acute malnutrition. So we turn to a technology that we've developed over the years, which is to be able to take a snapshot of a person's microbiome in time and then replicate their microbiomes in the guts of mice that have been reared under sterile conditions and are now colonized just with a human donor's microbiota. So we can replicate in many mice the donor's microbiota. We can follow the transmission of these microbes across generations of mice. We can also humanize them in another way, which is to give them the very human diets of the donors of these microbial communities. So we can look at diet by microbiota interactions. And by the way, that's an interesting way of exploring next generation innovative foods, designing food from the inside out and thinking about the microbiota as a metabolic organ that processes food and gives us food in the form of a metabolic output that sponsors healthy growth. But these types of transplant experiments that I've just described can be used to ask how much of an individual's biological feature is ascribable to their microbiota. Can we transmit part of your UNIS to a mouse via your microbiota? And that's exactly what is done with these discordant twin pairs. And the results are shown in this somewhat complicated slide, and I'd have you look to the left and notice in the upper left-hand corner a graph where there are lines that are colored red, which represent the weights of mice after they received a transplant of a gut community from a healthy co-twin in one of these discordant pairs. And we're looking at weight over time as a function of diet because we're giving the mice the very diets that are being consumed by the donors of the gut communities. So first the mice receive a Malawian diet, and the recipients of the healthy gut communities maintain weight. But the recipients of the kwashiorkor microbiota lose weight dramatically, have all sorts of metabolic derangements. There is a hit on their immune system. You give them RUTF, and there's a transient improvement in the microbial community performance of the mice that have received the malnourished child's microbial community, but it's not durable, just as we saw in the humans themselves. So the effects are not durable. They are dependent upon diet and microbial community interactions because we don't see these abnormalities when we give the recipients of the malnourished child's microbiota a healthy diet. So here's the problem. We have a microbial community that is not functioning in a particular dietary context sufficiently to support health, at least in the recipient mice. There's a causal relationship between the microbiota and health. How are we going to repair, in a durable way, the deficiencies in the microbial communities of these children? Are we going to have to give them food, therapeutic food, for a longer period of time? New therapeutic foods? Are we going to have to give them microbes themselves to help sponsor reformation of a healthy gut community? We are using methods to identify the very microbes that are necessary to promote healthy assembly and functioning of a gut microbiota. We're trying to deal with the question of, can you have normal physiology, normal metabolism, normal immunity, if as a child you're walking around with an immature organ in your body? This is a microbial organ, the gut microbiota, that is not properly formed. How does this immaturity and poor diet contribute to the abnormalities associated with malnutrition, impaired growth, immune dysfunction, the neurodevelopmental problems? The brain at birth is 12% of body weight. It's incredibly demanding in terms of energy. As adults, it's only 2%. What happens if the microbiota is not functioning properly? And what's the impact of maternal undernutrition on the transfer of microbes in the assembly process? So we have a dream and a vision that I'm sharing with you in this last slide. A more comprehensive and complete view of normal human development that has a microbial component. We may be able to refashion these aspects of development more readily than other aspects of our development because we can manipulate the microbiota. We're targeting a population of infants and children who are at elevated risk for stunting and malnutrition. We're trying to improve therapy by developing new generations of foods and new ways of administering microbes that are naturally occurring, that are part of this normal program of assembly to individuals at early phases of their lives. We want to make this intervention that's durable and generalizable and something that could be used beyond treatment to prevention. That is our hope. That is our dream. Thank you.